Go when you start. We get started.
All right. So, welcome to the first panel discussion session for this workshop. This is titled Brain Behavior and Synchronization. So, I will be the moderator. I'm R.D. Lila from the University of Washington. And we have four panelists today. Dr. Kass from Carnegie Mellon, Dr. Ma from Emory, Dr. Park from University of Toronto, and Dr. Ali Shojai, who will be joining remotely from University of Washington. Okay. So this is pretty much the information I was given to come up with a few questions. Other than these need to be tough questions. Okay. So I was wondering what's special about these four panelists. And if you look at the list long enough, you realize that I think what's special is, or one of the things that's special is that they worked on a broad spectrum of neuroimaging data. And what I mean specifically is things going from neuronal level data through MEG, EEG data, all the way to fMRI data. So the focus will be on trying to see how we can possibly integrate these different, what somewhat are perceived as different communities. So one way to categorize neuroimaging data is into three clusters that are sort of microscale data, where you can sort of appreciate neuronal level, or basically neurons. And of course, you've zoomed in a lot, so you lose a bit the big picture. What's in between, maybe mesoscale data, so something that tries to recover a bit the big picture, so basically clusters of neurons and some long-range interactions, and then macroscale data. And the macroscale data, everyone can see, all of a sudden we recover the brain, so it's easy to see that that's a brain. But of course, we zoomed out a lot, so those are not even clear. Those are bundles of neuronal connections now. These are all structural connectivity data. I don't mean to focus on structural connectivity data, but we can sort of project this categorization onto functional connectivity data. And here on the left-hand side, we see calcium imaging, where that's supposed to be a video, but it's not super easy to see, but where you can basically appreciate spikes, so things that happen on a millisecond scale. And then you go on to things that are a bit more macroscopic, things like local field potentials, where now the spikes are averaged out all the way to EEG and MEG, but you still have good temporal resolution, all the way to fMRI, where now you have great spatial resolution, but you have pretty bad temporal resolution. Also, a lot of things going on there. So you raise your hand, you see a peak that comes in 5 to 9 seconds, maybe. Okay. And this takes us to the first questions. The first question is, how do we go from micro to macro connectomic? In particular, how can we integrate micro, meso, and macro scale brain connectivity data to better understand brain function? And one example is identify cognitive and behavioral differences. So the focus can be anything really. It can be on experiments, can be on neuroimaging technologies you would like to have, statistical methodologies, or even just knowledge that can be carried forward from one to another. Okay. So we have three panelists. We'll start actually from Dr. Ma.
Hi everyone, my name is Tianwen. I'm a junior faculty at Emory University. Just seeing this question, I just feel like it's a really broad question for me. And then I just realized that maybe integrating different scale brain connectivity, it just reminds me of an example that I recently got to talk because I work with a lot with like a neurosurgeon at Emory, and they were actually talking about doing like epilepsy surgery. And I know like EEG is known to identify and localizing the source of the epilepsy, but they sometimes, and also the recent research started to thinking that epilepsy is like a disease of network. And then maybe we need to bring more modalities of the kind of the other type of imaging modality before surgery and intraoperative and also post-surgery, because they would imagine that with a successful epilepsy surgery, and they would expect to see the alteration in like both structural connectivity and also the functional connectivity. That's like something that I thought about.
Okay. My name is Jun, and I hope I'm not the only one who got confused by the this kind of slide that they already posted. Like I saw like your faces and it's all to like a word exactly like me when I first got it. But I'm working more on the fMRI side, and then I'm cross-appointed with psychology, so I had chances to work with collaborators there. And when I try to identify the association between functional connectivity to behavior, or more generally in your imaging to cognition or a behavior, I stick to focus on the behavior, not the not the brain disorder prediction. For example, the one that you can obtain from the human cognition project. And I think it is Wes who tried to examine the predicted behavior of the functional connectivity on the behavior. And if I remember correctly, the maximum R squared that can be obtained using the functional connectivity measure is at most 40%, right? So if you're really interested in building a predictive model for the behavior, then should we really try a more deeper model to improve 1% of the accuracy, or should we improve should try a better to improve the quality of the data? Because everyone knows that resting state function connectivity, it has some it has lower test retest reliability compared to the structural connectivity or something, or some other measures derived from the DTI or SMRI. And there are tons of issues that need to be resolved through regarding pre-processing, high dimensionality, and everything. So probably identifying some of the missing information from the functional connectivity would be better in studying brain behavior associations than trying to use fancy machine learning models to improve predictive performance by 1%.
You're an online person?
Um,
Don't you have someone online who's
yeah, he's not here yet.
I have to talk now.
So I had the same. I guess we all had the same reaction, which is we really. I mean, I feel like if anything, my reaction was more extreme. Like I did not get at all what was being asked here, and I had email about this, trying to figure it out.
I neither you explained it.
Yeah, and I didn't get an answer. So,
Could I add something before your talk?
sure.
Yeah. I think the question is mainly come from actually MH as an initiative is about this topic of brain behavior and synchronizations. Okay, and actually
It comes from what?
the AIMH.
Oh,
This is a brain initiative has a program recently in last 3 years. There's a specific program about brain behavior and synchronization. And actually one of the critical things, they are trying to develop the experimental paradigms to measure the multiple information simultaneously. And they wanted to measure the neurons and also the brain functions, and also they using the videos because neuron is the mice running around, and also they record the videos, and basically they simultaneously record all the information and try to link all the things together. That's basically where it come from. Okay.
I mean, what can I tell you? My honest reaction is, people want to do all kinds of stuff. It sounds cool. It doesn't mean it's a good idea. Honestly, the way I think about this kind of thing is I believe it's fair to say that all the work that we do is abstracted away from the actual problem. I mean, we solve something. It starts, but it always starts with a real scientific problem, and it's very specific. In our case, brain regions. Specific kinds of things that we try to think of, that questions that need to be answered, and then ultimately come up with some set of methodologies that are supposed to be more general than that, right? But when you ask any of these questions, it's like, I don't want to answer any of this in the abstract. I want to know, okay, what specific problem do you want to know about? And so what I end up doing when I read different papers in neuroscience in all the different areas, because you kind of have to, I read neuroimaging papers sometimes, even though I'm not an expert on that, but I read them, and they're all providing interesting information. But to me, this is all actually at a very qualitative level. And you see certain things like take a specific any of the major diseases that get studied, say in mental health or addiction, which is almost mental health, and you see them studied at different levels with different technologies. But what's interesting to me is that you see results that have some semblance of consistency in different ways you look at the thing. Like a particular brain area becomes important, but you see it in the neurons, individually, and you see it in the the EEG. There's something that seems relevant to something that's going on. You see it in fMRI. It's a much bigger area of the brain than we would ever look at, but still, there's something that's consistent, and that's really nice. But I cannot imagine how you would deal with that statistic, or why you would want to deal with that statistic.
I think because the current the status is because usually we do experiments independently for all three different levels.
No, I get that. No, I get that. Believe me, I understand what multimodal inference is. But and there are places where it makes sense, right? Certainly, I mean, Hernando asked about LFP and spike trains. Sure, there's definitely cases where that's going to make sense. And people had used MEG to try to, or I'm going backwards. Maybe it's I don't even remember. This is terrible. Which direction it went, but you use it's probably an MR or maybe functional MR to do something that's going to sort of set you up for your MEG, or something like that. So there's places where it makes perfect sense, but it's like it reminds me of I was on CATS, a Committee for Applied and Theoretical Statistics at the National Research Council. It's run by the National Academy of Sciences, and I was on that for a while. And one of the things that was happening when I was on it, which is quite a while ago, is that you get these people, they're all coming in from government, and you get, some part of the government. And what sticks in my mind is fisheries. And they like, well, we have all this video data, and we have all this, and it's like, what do we do with the data? And it's like, well, geez, I would have thought about it. So you collected all this data without a goal? I mean, it's odd to me. It's just, put it this way. I'm not saying it's bad. I'm not saying it's wrong. I don't want to be misinterpreted. All I know is I don't know how to think about it.
No, I think
I have to start with the problem, and it has to be really specific, and then we can go more abstract.
Yeah, I completely agree with you on that aspect. Actually, in the last 10 or 20 years about the brain initiative, one of the kind of key emphasis is they try to simultaneously measure multiple information across multiple scales simultaneously, develop the device for doing that. And I can give you one example. My friends basically, what they want to do is simultaneously acquire the function of mind and the neuron data simultaneously, and see how does this brain connectivity. They want to provide neural basis for the functional connectivity.
Of course.
That's one of the key things. Another thing is basically they want to link the functional connectivity or with the specific the disease type, like the mental health. Why the different people, is behaving differently. They wanted to understand this mechanism. That's one of the key reasons they try to do that. Nobody knows how to do it because there's no data connected, even though have the device. Sometimes at the beginning, people think of that way.
Okay, so I searched. Are we gotten to synchrony yet? Are we jumping? Can I jump ahead to
you
it?
can
Yeah. So when I got this, when we had the email exchange,
Yeah.
and I still didn't know what was going on. I just started searching for synchrony in neuroscience, and because I know about, I worked on synchrony for pairs of neurons a long time ago, and it has a very there's a thing that people used to talk about and blah blah blah. It's an interesting subject there, but then I looked at what else is what are they doing in neuroimaging? What are they? And what kept popping up was social synchrony across subjects. This is the big thing that if Google immediately gives you. Is there's tons of papers about you image groups of people at the same time. It's mostly in EEG. But sometimes they'll do scanners with two people. I don't know how they do it, or they do it like they give them exactly the same thing, and there's I don't know. I don't really know what else they could do. But the one that caught my attention was this EEG with groups of people, and they want them at drug. But what I immediately think of is like, well, there's two obvious things going on. There's behavior. Like, why are these, what they're finding is these subjects they're responding kind of in sync with each other, right? Under certain circumstances. There's a discussion, people are talking, and they've synced up, right? According to certain frequencies and I'm like, well, they're talking to each other. They're having certain behaviors. Part of it's related to attention. And it's like, okay, there's certain behaviors that are driving things, and there's attention effects. And it's like, what exactly do you want to know that's more than what I already told you? We know there's a behavior going on, and we know that there's similar behaviors. We know that they're paying attention at the same time. And that's that could be driving everything you're seeing. What's the question? What do you want to know? And this was when I read a few papers just in case I could, and it's like I couldn't understand what these people wanted to know. They think that it's a scientific finding to show that a bunch of people synced up. And it's like, okay, good for you. You got your paper in PNAS because it sounds like really cool. But I don't understand where the science is. I really don't. So that's what worries me. And NIMH can be fooled like anybody else. I had a great experience recently. I was on what turned out to be a really interesting panel. If I can monopolize for a second, an anecdote. It's interesting. So I got asked to be on this committee which I can now talk about because the report was released. And it was called, actually I don't remember the title of it, but it was like how to guidelines for big data in mental health. And there were, I don't know how many people, let's say 20 scientists. And I believe I was the only PhD in statistics, but there were a few other people who clearly did statistics for a living in some form. And a lot of them were geneticists and other people who were quite quantitative. And I have to tell you, there were periods of time when I was worried about whether I could sign my name to the document that was going to be produced. But in the end, I was happy with it. I think it's a good report. But a lot of these very, very basic issues come up. And, it takes a lot of hashing out until you end up with this place where you're just trying to tell people, don't do crazy stuff. And they have to be instructed. It's I made a remark about how I wish that people understood that there's no universal context-free measure of evidence, statistical evidence. It's like you always have to know what's the context. A p-value doesn't mean the same thing in this case versus that case. It's got the same mathematical definition, but the relevance for the science depends on the science and the way the experiment's done and a bunch of other things. And so there's these very, very, very basic points, and I felt like that's the stuff that somehow we're not getting across. And I guess I'm just saying, it feels like the same. NIMH may well be doing this. I mean, it obviously must be. But it just, I'd have to be convinced that there's real substance there. It's hard to imagine there isn't, because there's usually very smart people who are behind this. It's just that it's so easy to get caught up in something that sounds really cool and that uses, I've had so many different people in my working life in this area come up to me and say, well, you can take statistics and find this. And I'm like, no, I'm not going to be able to find, but how do you think this magic works?
I think you make a great point. I think in the last 20 or 30 years, if you're looking at the grants funded by MH, they connected tons of the neuroimaging data, and also all the neuron spectrum, all the other data sets, and they put it on these kinds of centralized resources, and all these studies are quite independent. Also, they have all the behavioral data, right? Or imaging genetics, right? Or they connect all these kinds of data, and actually these days basically put all these information together. But how much the progress NIH make in the last 40 years is still the definition of the mental health. Most of mental health disease is not well defined, right? And then the next question is, how do you really integrate all the information together when you basically carry out a totally different study? It seems like they are not relevant, but actually, they certain degree they also related. Then they think about one thing they have concern is because this data are connected to different subjects. And basically, they are not behaving they're supposed to, and also larger variation across the subjects. And why don't we develop devices to collect the data simultaneously and looking at them and find their relationship? That's the basic. I think that's the one of the key motivation for this kind of program. Basically, try to push forward.
Maybe if I can add something. So a more concrete problem, I've worked a little bit with MEG data and fMRI data, both trying to get something like, oh, maybe connectivity is something that varies over time. If you do it in fMRI, you gotta be a believer to trust. At some point, you gotta be a believer to trust that you can what you're truly getting there is true variation. It's not just random noise that you are fitting just because you're taking a smaller window, so you're observing just sample variability smoothed out. And then you go into the MEG. It's exciting. You got hundreds of thousands of observations for very few mistakes. And it's like, oh, this is exciting. Let's work on this. And then you start doing reconstruction, so trying to map those signals on the cortex. And there you lose faith. So I have found sometimes myself in the past being maybe there is something to be learned from fMRI that could inform MEG reconstruction, and then all of a sudden you could use MEG reconstruction temporal resolution to inform those to have an idea of where the connectivity happens. So I think that's one example of
I agree. I'm familiar with that because we tried to work on MEG for a while too and abandoned it. But we did no. I should say, but we did a couple. We have some I think actually quite good studies with MEG that we did, and I was really happy with it. I think they're super interesting, and so like I'm familiar with it, and that's exactly one of the cases I was trying to remember how this works. But it's pretty much what you said, and I think there's many people who've done that kind of thing. So that's certainly that's a good use of
actually
two
looked it up,
different modes.
and there are people who have been able. I don't know MEG. There's a magnet involved, so I'm not sure how that interacts with an MRI machine. But there are people who've done that at the level of EEG data. Although this doesn't seem to have gotten a lot of traction, so this I don't know why, but yeah. So measurements at the same time, EEG data and fMRI.
It's absolutely being done. There's definitely people who've done it.
There are issues apparently with the electrodes warming up and then noise.
Well, you have to be careful what kind of electrodes and all that. I mean, my friend Emory Brown, there was a while when he spent a long time working out how to do exactly that in the, and I don't actually know what became of it. I haven't heard about him talking about that recently, so I think he did something and then stopped. But yeah, people do it. Yeah, it's a good example.
So that's one of the, okay, thanks.
Another thing is basically in the neuropsychiatry. And these days, people think about basically using the brain imaging-related biomarkers, and basically using that to define the disease, subtype of disease, and also they try to develop intervention to treat this type of the brain biomarkers, and then you basically using this is basically the connections on that to link with the disease type. And also, basically, they want to basically do this type of analysis mainly because they want to understand the neural foundation of these brain biomarkers. That's the basic. Another thing we can think about that way is synchronization. Basically, that synchronize the information across.
Maybe another related aspect is, I think it's important to understand in some sense what is the atomic functional level at which the brain works, right? And I do wonder if that's the neuron or that's a cluster of neurons. When we work with sort of fMRI, it feels like we're working with clusters of hundreds of thousands of neurons that we averaged out and captured the fMRI signal. That's pretty much maybe how many neurons there are in a millimeter cube of the brain, which is the voxel size. And then I wonder, are we losing something by averaging those out? Maybe not. Maybe neurons working, it's not important to tease out the contribution of every single neuron in those 100,000, but it's hard to believe that we should represent 100,000 neurons as one neuron. So I wonder how down that, how much we should zoom in. Is it really neuronal level or is it a bunch of neurons? I don't know.
I thought this problem from a little different angle. For example, if you think about the functional connectivity is the currently defined as a correlation between two regions time series. But if you think carefully about the regions time series, it's kind of average, right? You define a region, then you kind of average, and then you take a correlation, right? But if you just look at the like a voxel level, like time series, and try to quantify the dependencies between like two regions, then the problem becomes a little more different. For example, averaging means that you're assigning the equal weight to each voxel for every same participant, for example. And can we like assign like different weights or like more like a personalized weight to each different individual so that we can maximize the definitions of the like I mean, you can benefit from a better definitions of the functional connectivity, right? For example, like some person may have more localized like sub areas that are more connected to the others, like compared to the others, right? But if currently like we are just like averaging everything and then computing the Pearson correlation, that's it. And they're all like if you like, view this problem more from a VAR perspective, there are like all the like interdependencies like within like a region. So like there's a like complex temporal process and there is a complex spatial process. But if we lose so much information by averaging out. And if you think this problem from a dynamic functional connectivity, there's a non-stationary structure. So if you're only interested in like static connectivity, not about the like effective connectivity, then like those kind of like time varyingness is just adding a noise, right? Like so that's why like I'm kind of like I'm thinking that like there is some like room for development like by looking at the more kind of like micro scale like a functional connectivity, not at the like a neuronal levels, but still at the like voxel level. We are like I mean, because like functional connectivity is a standard like statistical measure. It's not like you kind of measure like how thick your like cortical thickness is, right? So like we need to try our best to reduce those kind of like unwanted variations, and from that perspective, like we are losing too much information by trying to be like, simpler. Yes.
So another thing I'm generally not very comfortable with is things that rely entirely on averaging across subjects, for the reasons that you've articulated, and I think a lot of everybody in the room probably appreciates. But you know, in the studies where I've been involved, where there's multiple subjects, I pretty much always say, show me some individual subject data. I want to see that we see it in individuals. It doesn't have to be every subject. Because one of the things is, you know, subjects are so different. And when you go down to the neuronal level, there's no reason to think that people have the same detailed wiring. What they have, from mouse to mouse, for example, is the different areas that humans have identified as parts of the, let's say, visual system. Those are consistently connected with each other in a certain way across mice. But the details of how that's done, the number of neurons, the way you've got, don't forget, you've got inhibitory and excitatory neurons, you've got different layers of the cortex. Like these are all details that are really important. And you're never, you know, you don't get that with any of the neuroimaging techniques. So when you ask something like, you know, what level is important for understanding, I mean, they're all important. But you know, behavior emerges from this immensely complicated system, and then it becomes a matter. I mean, if you say what's the most important level, it's like that's just a matter of taste. I mean, obviously I like the most detailed level because it's just the way I think. And it doesn't mean it's right. It's just like to me that's the most interesting part. But I mean, the other part is interesting too. You know, fMRI. Like we wouldn't be where we are in neuroscience if it weren't for fMRI. Like there's no doubt in my mind. Like there's been a lot of really interesting studies in fMRI, so we need it all. My only question is I don't see the value. There's some things that I don't think need to be formalized in statistics. I've always felt this way. I don't think there's a lot of meta-analysis where I don't see the point of formalizing this stuff. You know, I'd rather do it informally. I'd rather do it step one by one by one by one. Let's analyze this, let's analyze this, let's analyze this, and then we come to more like a consensus or whatever. It doesn't have to be a statistical formula that tells you how to do it.
To add, so precision neuroimaging is a new thing now. Like, so more people are doing exactly that. They say, forget about trying to do a joined up analysis. I've just got 100 hours of data on this subject. I got 100 hours of data on that subject. And yeah, some people are taking that approach.
Any comments from the audience, by the way? Do you want to add something, Tianwen?
Yeah, I think like you were talking about like doing the joint analysis of integrating all the kind of modalities. of course, this is very interesting and also very meaningful, but from a clinical perspective, like a healthcare perspective, we probably need to talk about the budget. Like if you want to study the disease or if you want to follow the disease progression of a particular patient, of course, doing a functional MRI may review something very insightful, but it might be time consuming and money consuming. And I think right now, for example, I think when I was in SFN last year, I noticed that I think it's Luis Sanchez from Johns Hopkins. Like he is actually replicating the glucose metabolism of the brain using the EEG measurement because of course he's to study the dementia, the type of dementia. And for example, if you want to diagnose with Alzheimer's disease, then perhaps like using the PET scan is the gold standard to kind of record the concentration of the tau protein, but it's really expensive. It's like $5,000 per scan. But if you can replicate with like a relatively cheap and non-invasive technique, for example, the MEG or the EEG, and then they can still reconstruct the kind of the concentration of the glucose metabolism. And I think in that sense, it will still benefit the healthcare efficacy of the patient, and also because these kind of modalities very fuzzy, and it definitely motivates us to develop some statistical methodology to kind of reconstruct and compare it to the gold standard.
Yeah, I think my point is not just doing joint modeling together, rather than basically when you first see such a data set, it's quite unique because you simultaneously acquire them simultaneously. Then basically the within subject variation much lower than cross subject variation. You could be able to establish their relationship in certain degree. We never understand that before. That's the key point. That's why people care about these kind of things. I think that there will be such a data set available to the researchers pretty soon.
Yeah, I really like Robert's comment. So I can see that the science development starts from you have very specific questions that. So I used to talk with people working on big data. They actually see that recently the research has shifted. So it's no longer like you have a very specific questions, and then you do experiments and then you collect the data. He says that the research should be you generate a lot of data and then data kind of create the question specific hypothesis. So that's I think that's a trend nowadays. Even and even though I realize the collaborator I work with typically still start from a very specific scientific question. And since Robert mentioned that, I'm very curious. Since you have already worked on MEG, so why you move away from it? Would you elaborate on any scientific insights?
I think it feels in retrospect somewhat coincidental. I had one student who did these really interesting studies that we published, one in particular, but she did several papers. And then I had another student who was going to work on that, and we got to a point where the investigator, the person we were collaborating with, who's somebody I like very much, and she's very successful, but I'm about to say something negative. She thought we had publishable results, and both the student, who was really a good student, and I said, no, we don't. We're not going to publish this, because it just, it didn't pass our standard. I mean, sure, we had some decent p-values somewhere, but we didn't believe them necessarily because of all the stuff we did, right? And so then I didn't have another student who said, oh, I want to work on if some student had come along and said, oh, I really want to do MEG, it might have happened again. But we had that happen, and then I had another student I did supervise for a project, because at Carnegie Mellon we have a year-long project in data analysis everybody has to do. And I had one student who did one of these whole brain things, and the guy who's again a respected scientist I like very much, I've talked to him many times, but he really had this idea that again the data should tell you what's going on, right? He had this very not specific, open, and this guy was again a very good student. He worked on it for this whole year, and he had nothing but negative results if you're rigorous. if you're rigorous, right? And so, that's what happened. I'm not saying that MEG is not interesting and important. I'm just saying that's what happened to me.
Any questions or comments from the audience? Yeah.
So just from experience, I'm firmly in Robert's camp on this multimodality stuff. And with reference to clinicians, I've had a lot of clinicians that I work with say, we want to do multimodality. We want to look at all these different multiple modalities and put them together. You say, well, what do you want to do with that? What do you want to answer there? And they don't know. And so you're starting, let's say you're starting with fMRI, you're in N by N right now. And then you're going to do connectivity. Now it's N by N by N by N. Then you add in some other modalities and the dimension just explodes. So like if you don't have a pathway for the questions you want to ask, you're just going to get in trouble.
So I think one of the other questions is that NIH is actually pushing for all these big studies, right? So we moved from a one-site study, and what Tom was saying before, it's wonderful that you can take a subject and analyze their data or collect their data 100 times. But for example, in PET imaging, that's not possible. And the NIH is pushing for these big initiatives, and all the collaborators and somebody that I can relate to what you're saying before. People just go from simple clinical questions to just digging into the data and finding answers. So how we as statisticians contribute or somehow, convey to the NIH, sometimes some of these things are not actually the best. How do we get a seat at the table? Because I think that's the question.
Well, I think there's actually a lot of seats at the table. I think it's a matter of talking to people. Honestly, if it's an IMH, for example, now they contacted me, but I mean, I've talked to the certain program officers for years, and I think they really want statistical help, right? I think they know that they want it. So there's a sense in which there's going to be seats at the table. It's just a matter of finding them. And I think you probably it's like what I tell people who want to get into any of this research, the younger people, that you got to reach them at their level first. You have to help them on something that gets them going. I always say, like, if they want to do a t-test, like, do a t-test. Just get them going and gain their confidence and have a conversation, and then move slowly to the deeper things, and hopefully you build up trust and all that. I don't think you can jump in and tell somebody they're full of shit. It's like it's
right. But
I agree with you that there is a move in those directions.
And I think a lot of the things are happening because all these RFAs are published after they have been discussed, right? We figure out, okay, so here are the announcements for the grants, and it's mostly going towards major multi-site studies and data integration. I think a lot of the...
But you can speak to this better than I can. I mean, you're involved in one, and I bet it's a good study.
Yeah, I mean, at some point, it's sort of irrational for the NIH to fund 100 studies of Alzheimer's of 30 people compared to one with 3,000, right? Because you're going to have like 100 underpowered studies. You have to do some sort of meta-analysis to get something, and it's just and there's no sort of mechanism. The NIH doesn't give money to sort of combine the data sets in any way. That's their shortcoming. That's what I tell them all the time, is that you should pour in money to combine these different things to make a mega study out of them. But it's not from a taxpayer point of view, that's completely irrational, right?
And related to that, I would say the incentive structure, in universities is getting kind of out of whack, but it's really bad in medicine. I mean, again,
the whole bonus.
Chris, I think you already have some.
I just wanted to say one thing that Rob said is super important, and that's like, it's got to be a conversation. It can't be you lecturing at people. People don't like to be lectured at. It should be a conversation of equals, of collaborators. It can't be you're doing your science wrong, you're a terrible scientist. It has to be we're coming at this with the same goal, which is to improve the science that we're doing. Otherwise, they're just not going to listen to you.
Yeah, and again, like the people who I'm negative to are people I respect deeply. I just want to make that clear. It's just that they don't know. Like one of my really closest friends wanted me to do something with it's a lower level, much lower level study on mice and stuff, and she said something about how many mice and she's got and I said, and at some point I said, well, I don't think you have a large enough group of mice here. And she said, well, that's the standard in the field. And I said to her, I don't think your field knows what it's talking about, honestly. But I knew her well enough, and she had already been a close friend of mine that I could say that.
You have to give them the respect to speak your mind.
I think all the discussions are great, fruitful. I think if we're looking at AIMH, all the studies funded the last 50 years, and we see a trend. I personally, saw a big trend nowadays. Okay, usually we just run multiple studies with a small number of sample size. Right? These days, basically, sample size is getting larger, and also the older days, the data is controlled by all each PIs. Okay, and put it in their pocket, never share with outside. Right? These days, I say, and you have to share. Okay, that's great because there's a lot of things you can do with that. Okay, and another thing these days, basically, AIMH try to centralize all the data, put it in a central space, center. Basically, you can download all the data, and after you fulfilled all these requirements.
Comments on that?
The problem is nowadays.
We don't want to get into those comments.
Nowadays even ABCD, it's pretty messy.
Well, they're not doing it anymore.
ABCD is withdrawing from NDA, so.
Yeah.
Wait! I didn't hear. I want to hear this. What? Make this comment.
They put on. Okay, so yeah, so.
I mean, I can hear you, but.
So the next release of ABCD, which will be in sometime this fall, is going to be done not through NDA. We're divorcing ourselves from NDA going forward. So.
Wait, explain what that is.
The NIMH Data Archive, that's the central repository I think that I was talking about. Yeah.
So wait, so ABCD is gonna do it just completely separate.
It's completely separate.
Because, can you say why?
Because they put too onerous of a burden on us to do stuff that was bullshit and wasn't helping with the data release at all. It was just like it was thousands of hours of person work really resulting in a worse data set. So we just complained, and the ABCD is by far the largest data set on NDA, and so we said we're just not going to do it anymore. So, and NIDA supported us basically.
Yeah, the aging is also divorcing themselves.
Yeah, and HBCD is also going to do it.
Let me make one more comment, just sort of in defense of the whole way people operate, generally speaking, and that is that one thing that I feel about certainly neurophysiologists, and I think it's true of all experimenters, but neurophysiologists in particular, is they're very opportunistic about technology. In other words, there's something that new technology, and it's things like the electrodes. This neuropixel thing is like a really big deal. That's a good example where people suddenly start to say to themselves, or calcium imaging just wasn't that long ago. There's many things that come in. They're pretty new technologies, and the experimenters are saying, oh, that sounds like people are. That paper's in Nature. It used this. Oh, that's really interesting, and they're asking themselves, okay, here's a new tool. How can I use it, right? And so there's a great natural tendency. And by the way, statisticians are the same, right? We do exactly the same thing in our field. So that's a good thing. It's just that we're talking about they're using a new tool, and the problem is when it gets into data analysis, they think a screwdriver is a hammer. Right? And they're trying to, it's just, no, you want a hammer, you don't want to, it's like they just don't, they're at the wrong starting point. So, yeah, I don't know. I just think my opinion is we got to keep trying.
Yeah. Can I say that's one of the things that really excites me about MRI is that every 2 to 5 years they come up with some new acquisition, some new properties. Diffusion is a good example in the last 10 years. It's amazing what you can do now. So that's cool, but it's true. Like you've got to engage with them to then develop the appropriate statistical models with that data.
There's a question there.
Yeah, so this is back to the original question that was posed. But I was thinking about ways in which I might want to address this or that I would find useful for someone to address it. And I know that there's a lot of question around what is functional connectivity, like what is actually happening in the brain that leads to this observation. And one point of contention I've seen is that is it actually even about neural activity or is it about other cells in the brain like astrocytes or glia and their demands for oxygen to continue to operate? So I think it would be interesting to use, like within a person, if you could have measures of neuronal and maybe even other cellular activity in conjunction, like at the same time with the bold signal, that might help us disentangle what the cellular basis of this effect is.
Thanks, that's a great point.
I mean, there have been some studies along those lines, right? There's people who have done not in humans. There was a really cool thing that this guy named Logothetis did years ago, where it seemed very careful to me, where he was comparing LFP to bold signal, and it was an heroic effort as far as I could tell. It was like very challenging, but he did it, and found that there were, to me, it helped me think that Bold was finding a fair amount of good stuff because it was there was a pretty high correlations in the way he did it. And another guy who I know who's done some of this it's a completely different thing, but just you can track. I'm not sure. I don't remember how it works. Glucose metabolism, again, like in a non-human, right? It's invasive, but you can see what's happening to the glucose. There's ways of doing that, and that's informative then too, right? So I mean, there are ways of attacking that, and I think it's being done, but I don't know. Again, at a high level of like, if I were in charge of NIH, how much resources going into that versus this versus something else. Like I often wonder. I can't imagine trying to figure that, the balance of resources at a high level for research, because there are so many interesting things that come up.
Always there, the studies that you mentioned is mostly based on the animals, right? For human neuroimaging, we mostly care about non-invasive techniques, so there's kind of technical barriers at this moment, right?
Yes, but to me, what's really cool is when you see things that happen across different species. Like, to me, that's really solid. When you see it a mouse is a lot different than a monkey, and a monkey's a lot closer to a human than a mouse is. But you see it across all three, and it feels like, okay, we're onto something. That's all I was trying to say.
You find a consistent evidence that they are related across monkeys, and then
there's certain things that are homologous. I mean, it's pretty gross, but yeah, I just think that's a plus in terms of the science. Because from a purely scientific point of view, it doesn't matter what, we're just trying to understand how it works. But of course I think personally that there will be, Tom was saying there's new acquisitions, technology all the time. I think there's going to be really, really cool neural signal methods that are non-invasive at some point. The problem is it's very hard to predict when. I think people are going to figure out some kind of nanotechnology thing that you can get into blood vessels and target a certain part of the brain, and suddenly you can see something. It's like it's going to be people have been talking another different thing about voltage imaging for quite a while. It still isn't really effective yet in details, but voltage imaging, if they can finally get that to work well, that's a big deal, because we go from neuron activity to synapse and knowing which neurons are connected to which other ones when they're active. Like when you show that calcium imaging thing, you don't know which ones are connected to which other neurons. You still have that problem. But with voltage, you can track it. And so, that's going to be, to me, there's a number of technology things that will change things, but I don't know when they're coming is the problem.
Yeah, that's a great answer, actually. Thanks.
What's question 2?
That's a great discussion.
I was actually wondering whether we should go into question 2. I hope it's not as triggering, but from neural synchrony to correlation, so it's actually related. So how can different notions of brain synchronization be incorporated into neuroimaging statistical analysis to enhance our understanding of cognitive processes and behavior? And it can be, I don't know, synchronization can be anything, right? It can be time synchronization, space synchronization, as in registration. whether local versus global, how we think of amplitude versus phase. We saw some examples, or phase versus frequency. Tianwen, you want to take it first?
Yeah, I think because my background is mainly from the EEG, and also it's like a task-based EEG, it's not like a resting state. So I think like people in the BCI area, they tend to just they just utilize the amplitude, but they kind of ignores the connectivity to do the prediction tasks. But I think the recent effort, I see some like publication that also look at the connectivity when they're doing different tasks, and it really has like a great like prediction accuracy. And also, I think based on the amplitude and phase, and they can be used to do the artifact rejection. I know like many of the right now there are some like online methods that can automatically detect this kind of artifact rejection, like you eye blinking and also like other type of movement, but just like extracting the face and try to kind of remove it.
So I'm still like, I think I was still thinking of this problem from the fMRI perspective only. And like I'm still continuing like a previous like support of like getting away from the like averaging. For example, like one way you can like think of is in the task-based fMRI studies, you expect there's also like people often study like dynamic functional connectivity, which has been discussed several times in today's talk, but there's also a some kind of expected changes in activation levels, right? In the very first level. But if you like think about how GLM is being constructed in fMRI, we kind of assume that the activation is kind of constant and try to measure the beta from the like linear regression model, right? And if we can like measure the like time varyingness of the beta, then and we can study like it's like an amplitude or like variability, then like it might provide additional information that is not really like captured in static activation. And actually that is like one of my like first work in like neuroimaging statistics that like with Mark. But yeah, but that kind of explains that like probably averaging out the information is gonna be causing a loss. And if you think about the integrating the the synchronizing the time, I thought it more like a problem of like can you capture like time varyingness in the activation and connectivity, and how do we integrate it in studying the behavior of the or like a promote the better understanding of the behavior. Yeah, but I haven't thought of in terms of the like integrating or synchronizing with other like data modalities.
I can add something just before. So I think Tom said it's exciting to see where MRI is going, like get sub-millimeters fMRI signal, possibly different layers of the cortex. I wonder up to what point it will make sense to talk about registration. What does even registration at a sub-millimeter level mean at that point? Can we even find where that sub-millimeter is on this subject is on another subject? So should we do away with registration at that point, or should we add a layer of uncertainty to registration? That's another example of something. New technology will bring you questions in terms of synchronization, even though that's a spatial synchronization. Dr. Kass?
Yeah. You said that well. I don't know that.
I mean, I think I can only repeat what I said before. Like to me, there are certain things that are homologous across individuals, across species too, but I mean across individuals, which is it's pretty gross. It's not fine details. So you can't expect, I mean, I think what you're getting at is there's going to be a limit to, like registration may be useful at a pretty gross level because the brain part that's a big chunk of tissue is going to be pretty similar in one person versus a different person. Pretty similar. But once you get it down into those, lower levels, more accurate, more smaller spatial scales, you just have to expect it's going to be different. It seems to me.
We've I don't know this song. A lot of registration is based on spatial features, right? So you take structural data and you normalize it. So it's not based on brain function per se. So it's very clear that there's a lot of residual differences in brain function even after a spatial registration. So what we've been doing is, and I think a lot of people are starting to look at, is doing secondary functional registration locally after the fact, because even though we sort of register the brain and they're grossly the same size, our amygdalas are a little out of whack, or our thalamuses are yours is elongated versus mine. And so basically, if you're treating a voxel as a feature or an entity that you want to average over subjects, you might be comparing apples to oranges, right? I mean, because you're not looking at the same thing across subjects. And I think that's so. There's a lot of sort of work now on trying to do more registration.
So it sounds like part of what you're saying is you get a better idea of what the piece of tissue is that's homologous when you look at function.
Yes.
In addition to the structure.
And I'll also say that it could also be that for certain things like the motor cortex, we're probably all the same, right? But if there's something that we have some behavioral task where you have a different strategy than me to solve in this problem, then all bets are off, right? There's no reason why our functions should look the same, right?
Okay, we have 1 more minute. Is there any final comments or questions?
Question 3.
No, there is no question 3. Okay, it was well timed then. Thanks again to our panelists. And thank you.
Yeah, good job.